Cut.
So today I will be talking to you about protein misfolding in disease. Protein folding is an extremely error-prone step in gene expression. So therefore the cell has many different mechanisms in order to help with this process. But because there are so many different mechanisms to help, there's so many opportunities for error. So I'll be talking about normal protein folding, misfolding, and then how this misfolding is directly related to neurodegenerative diseases, such as, or specifically, Alzheimer's. So first, I will be talking about the hierarchy of protein folding. First, we have our primary structure, and this is the chain of amino acids based on the mRNA that's translated through the ribosome. These amino acids have different R groups, which give different characteristics to the overall protein as a whole, which in turn represents the overall function of the protein. Now we have our secondary structure. So the primary structure will fold and interact through hydrogen bonds, as shown here in these blue dots. Some examples of secondary structures are alpha helices, beta sheets, and random coils. The tertiary structure, this is when multiple secondary structures start to interact together and we have a 3D conformation of the overall protein. The hydrophobic amino acids tend to stick towards the center of the protein to avoid water, while the hydrophilic tends to stay on the outside of the protein. And lastly, we have our quaternary structure, which is multiple tertiary structures coming together and interacting to form a larger protein structure. So in terms of the energetics of protein folding, so this can be described through something called the landscape theory, which is depicted through this energy surface or energy landscape. So in normal protein folding, there's not a single pathway that a protein takes to get to its native state, which is represented by the bottom of the funnel here, which is the lowest energy conformation. And so in order to get there, there's lots of local minima and maxima shown here. And so the landscape can be described as rugged. And so because there's not one pathway, the protein goes through these maxima and minima, but they have to overcome the kinetic barriers to get past the maxima and minima. So they kind of just work their way down and eventually to achieve their native conformation. So in order to help with this process, there are proteins called chaperones. Here we have a picture of this, the funnel that you saw just 2D. So here we have our native state, and we have chaperones, like I said, that help overcome those kinetic barriers to get past the maxima and minima. So here are shown fibrils and aggregates, which are the misfolded proteins. And so I know I said that native state is the lowest energy conformation, but that's not always necessarily true, because the whole structure function, the overall structure and function of the protein. So the structure won't necessarily be the lowest energy conformation in order to achieve the specific function that the protein needs to have. So, and then I'll talk later about these fibrils, which are just stacks of beta sheets, which is why there's such a low energy conformation. So when there's an increased level of stress in the cell, there's an increased level of chaperones to help reduce this stress and to help with the normal folding of proteins. And these chaperones are specific for which proteins they help fold. And how they work, well, how some of them work is they block the interactive surfaces of the misfolded proteins. So, like I said when explaining the tertiary structure of proteins, how the hydrophobic amino acids go towards the center. If for some reason the hydrophobic amino acids end up on the outside, the chaperones will recognize this and will bind to those hydrophobic residues and block the interactive surfaces, so they prevent aggregation or clumping together of those misfolded proteins.
Hey Kayla, can you really briefly define what causes stress?
So there's multiple different types. So if there's an overload of protein concentration and the proteins aren't being folded quickly enough, or if there's an overload of protein and they're all being misfolded and they're not being degraded fast enough, I'll kind of mention it in terms of the ER. I think it's the next here we have the endoplasmic reticulum and how it normally works. So the protein is translated, and it's translated into the ER, and there's modification and folding of the protein. If it's correctly folded, the protein will be encapsulated by a vesicle and then sent to the Golgi. However, if the protein is not folded correctly and misfolded, it'll be sent out of the ER to the ubiquitin proteasome system where it can be degraded. So this system is just a way to ensure that the proteins are correctly folded, and if they aren't, that they're degraded and just gone from the cell. So ER stress can be defined as when the ratio of the number of unfolded proteins is imbalanced with the ER's ability to fold the new proteins that are being translated into the ER. So with this imbalance, it causes stress, and with this stress becomes the activation of the unfolded protein response, or the UPR. So the UPR has 3 different pathways that it could follow. The first one I'll talk about is the ATF6, and so this is a transmembrane protein that gets sent to the Golgi, and the transmembrane and the ER lumen part of the protein is snipped off by SIP and S2P, which are just proteasomes, and it creates this ATF6N transcription factor, which then will activate and it will target these UPR target genes, which code for proteins that help in the folding of proteins. So it'll code for more chaperones, or it will help in the degradation of those misfolded proteins. There's the PERK pathway, where PERK becomes phosphorylated because it phosphorylates itself, and in turn it phosphorylates this transcription factor, EIF2 alpha. And so when this is activated, it in turn activates ATF4, which is another transcription factor, which will then code for chaperones or assisting in the degradation of those misfolded proteins. It also codes for GAD, which will cause cell death. So if it really gets overloaded, the cell will just go under the apoptosis. Next we have IRE1. And so IRE1 also becomes phosphorylated, and it initiates mRNA processing. And so what happens is this XBP1 will be cut in two places, and this intron will be taken out. These 2 parts will be ligated together by tRNA ligase, and it will create XBP1S. This transcription factor will then start, it'll bind to the UPR target genes and will also help in reducing the ER stress. Okay, so I've talked a little bit about aggregation and fibrils, but I'll make that clearer here. So when proteins misfold, they aggregate and they clump together because the hydrophobic residues tend to be sticky. And so misfolding can occur due to different environmental conditions, like changes in pH or temperature. They can happen through dominant negative mutations, and they can also happen through phosphorylation or glycation. So here's kind of just a schematic of how this process works. So we have our misfolded protein, and it can either be fixed through chaperones, or it can be degraded through that ubiquitin proteasome system that I mentioned earlier. If these 2 fail, then the misfolded proteins start to aggregate and form oligomers, which then order themselves into these elongated fibrils, and then these start to clump together and form large aggregates. And so later I'll be talking about these plaques that the aggregates form in terms of Alzheimer's disease. So energetics of misfolding. As compared to normal folding, it still has a very rugged landscape. However, the beginning of the protein folding process, if it's not being folded correctly, it happens really slowly because these conformations are so thermodynamically unstable and unfavorable. And so in an experiment with prion proteins, they found that protein folding was 1,000-fold slower when it was misfolding compared to native folding. So this, again, represents a super rugged landscape because the proteins are kind of getting through those maxima and minima in the funnel. And so this overall is described by the seeding nucleation model. And so there's two different phases of this model. There's the nucleation phase and the polymerization phase. So here in the nucleation phase, that's when it's happening really slowly because those protein conformations are so thermodynamically unfavorable. But then once the minimum oligomeric unit is made, so once there's enough of these here, the misfolding will just exponentially increase and it will create a lot of aggregates. So it's slowly folding, misfolding, and then it forms enough oligomers where it just takes off and forms a lot of aggregates. This here was from an experiment in a paper where they added the seeds, and the seed is considered that minimum oligomeric unit. And so they were just seeing if when they added seeds experimentally, if that would increase the aggregation rates, which it does. You can see it exponentially increases earlier than when no seeds are added. In terms of environmental conditions, so pH they found affects fibril formation and their morphology. So at a low pH of 2.5, branched fibrils were formed. So there was like one fibril in the middle, and then they were kind of branching off of each other. As to pH of 5.5, which really isn't that big of a difference, but they found that they were non-branched fibrils, and they were just long, like chains almost of fibrils. And so this just shows how it changes the morphology based on pH. In terms of temperature, compared to room temp, when they increased the temperature to 40 or 45 degrees, it caused a conformational change in amyloid beta from an alpha helix to a beta sheet conformation. I'll definitely be talking more about amyloid beta, but this is just to show you how temperature causes conformational changes. And so then, like I said before, these beta sheets have a high tendency to aggregate. So this is just a picture of these fibrils that I was talking about, and you can see how they're super ordered as the beta sheets are right on top of each other and right next to each other, which is why in that chaperone diagram it had the lowest energy state. Dominant negative mutations is another reason why protein misfolding occurs. And so these mutations are caused when there's a mutation in one allele that causes a structural change in the overall protein, and then this structural change causes a change of function. So there's a loss of normal function and a gain of toxic function in the overall protein. So if we have two monomers over here, and both do not have the mutation, then the protein overall is fully functional. If one was made without the mutation and one was made with, then the overall protein will be non-functional, and then when both have the mutation, then the overall protein is non-functional. So just some examples. For when insulin is mutated, it leads to proinsulin, which causes toxic function in B cells, and this is due to the mutation is addition or removal of cysteine groups, and so this causes incorrect pairing sites, which further leads to more misfolding. And then we've all heard of p53 in terms of cancer. There are 3 different types of domains, but the DVD, or the DNA binding domain, contains 90% of the mutations found in p53. And so when these mutations occur, the proteins aggregate, and these aggregates were found in tumor tissues. And lastly, we have phosphorylation and glycation. So we know the addition or removal of a phosphate group will change the overall conformation of protein. So when A beta is phosphorylated by protein kinase A, it causes a conformational change from an alpha helix to a beta sheet, similarly to how temperature affected this same protein. And then glycation is the addition of a sugar molecule to a protein, and so here I have a schematic of that. And glycation specifically, for example, affects collagen and elastin, which are age-related proteins. And so the glycation of A beta forms advanced glycation end products, and these ages induce cross-linking, so it causes less flexibility in the overall protein. So here's a picture of just the types of proteins, like the misfolded proteins that cause different diseases. I thought this was just interesting to see the overlap between the different diseases. I'm going to focus specifically on Alzheimer's, but as you see, like Parkinson's is caused by 3 different misfolded proteins. Alzheimer's has 2 of the same ones. So yeah, I just thought this was interesting to see the correlation between the different diseases. So what is Alzheimer's? It is an irreversible, progressive brain disorder that slowly destroys memory and thinking skills and eventually the ability to carry out the simplest tasks. So it's a loss in cognitive function. They can't really figure out like time and place. They have challenges in solving problems. There's memory loss that disrupts daily life. So it really affects the quality of life of the patient that has Alzheimer's. So here are PET imaging and MRI scans of a normal brain versus an Alzheimer's patient. And so as you can see in here, the red represents the amyloid beta protein. And so there's a lot more red in these scans compared to here. So yeah, it's just showing the differences. Okay, so beta amyloid is the protein that characterizes Alzheimer's. There's also the tau protein, but I'm going to focus more on this aspect of the disease. And so beta amyloid contains 39 to 42 amino acids, and it is made by the amyloid precursor protein, or the APP. So this protein can undergo two different pathways. There's the amyloidogenic pathway and the non-amyloidogenic pathway. So in the non-amyloidogenic pathway, which this just normally happens in your body, no toxic function, alpha secretase will bind to APP, which is a transmembrane protein, and it will cut the protein in this center domain here. Next, gamma secretase will come in, cut it again, and you get AICD and a P3. And both of these segments have no toxic function. They are completely normal in your body. However, for the amyloidogenic pathway, rather than having alpha secretase come in and cut the protein, the beta secretase comes in. And so there's like 50 different mutations, I think, that can occur on this protein. But the most common one is in position 717 from a valine to an isoleucine, which is why the alpha secretase cannot recognize the APP, so instead beta secretase comes in. And because this is a different enzyme, it's going to cut the protein in a different spot. So rather than cutting it in the middle of this red domain, it cuts in between this red and this section, and so we're left with APPS beta rather than APPS alpha. And on the bottom we have beta CTF compared to alpha CTF. And so just like normal, the gamma secretase will come in, cut it again, and then we have our normal AICD segment, but rather than P3, we have amyloid beta, which as you can see is slightly longer than P3, excuse me.
Do you know what part is the cytosol?
I believe.
Okay.
I could fact check though.
That's okay.
So what happens when these beta amyloid proteins are made? So this is a very similar schematic of what I just showed you, with the beta secretase and the gamma secretase come in. Here are the beta amyloid peptides, and they're being cleaved off of this protein, and they start to form oligomers, and then they organize into fibrils, and then they eventually form these huge plaques. And so this pink is one of your neurons, and pretend that there's a neuron above that plaque up there. And so if this plaque is forming and it's building and it's building, it's causing the cell-to-cell signaling is inhibited because the plaques are in the way. So because of these plaques, you're losing cell-to-cell signaling, and in turn, your body will also activate the immune response. And because this neuron isn't functioning correctly and it's not sending signals because it's not able to, your immune response will just come in and kill this neuron. So you're killing your cells in the brain, and you're losing cell-to-cell signaling, which is indicative of the loss of cognitive function in Alzheimer's disease. So this is the tau protein that I mentioned. I'm not going to spend too much time on this. So the plaques affect the brain on the outside of the neurons, while tau affects the inside of the neurons. So within your neurons you have microtubules, and your microtubules are stabilized by these tau proteins. However, in Alzheimer's, the tau protein becomes hyperphosphorylated, and so we know when you add a phosphate group, it's going to change the conformation of the protein. And due to this conformational change, the tau proteins are no longer able to stabilize your microtubules, and so they'll just start to fall apart and degrade because nothing's holding them together. And so when your microtubules fall apart and your tau proteins aren't stabilizing them, the wrong conformation of tau, they'll start to clump together similarly to the A-beta protein, and it'll form these neurofibrillary tangles, is what they call them, within the neuron. And so these tangles would just like clump up and your microtubules would just fall apart. So that's also a loss. So this will also activate your immune system and will kill your neurons because it's not functional anymore. So A-beta has many toxic mechanisms that specifically relate to Alzheimer's. These are a few of them, but I'm going to focus on receptor-mediated toxicity and oxidative stress. So here we have our presynaptic and our postsynaptic neuron. And here in black we have our N-methyl-D-aspartate receptors. And what these receptors normally do is when an action potential occurs during the depolarization phase, the magnesium on top of these receptors will pop off, and it will allow glutamate and glycine to bind. The receptor will then open, and it'll allow calcium to flow through into the postsynaptic neuron. However, in Alzheimer's, when A-beta is present, this protein ends up activating these extrasynaptic receptors instead. And I mean, these are still occurring, but these are also being activated. So when A-beta activates these, it allows the glutamate and the glycine to bind, and more calcium flows through. So there's this excessive amount of calcium being built up in the postsynaptic neuron, which leads to synaptic damage and eventually neuron death. So this extrasynaptic activity kind of just takes over the normal synaptic activity, and it causes neuron damage. So one drug that they have been using to prevent this is called memantine, and it inhibits these receptors. However, it has a moderate affinity for these receptors, and we want that because you still want calcium to flow through, you just don't want an excessive amount of calcium. So it will inhibit some of these receptors and hopefully these, and will just allow these to let calcium flow through during the depolarization phase. All right, oxidative stress. So A-beta contains metal binding sites in the middle of its structure, and it's able to bind copper 2+ and iron 3+, and reduce them to copper+ and iron 2+. And so because when this reduction happens, it's able to react with molecular oxygen and create a superoxide anion, which is this here. And so when it forms a superoxide anion, because of this, this lone electron, it's highly reactive with hydrogen, and so it forms a hydroxyl radical here. And so when A-beta is in this highly reactive radical form, it's able to extract protons from neighboring lipids or proteins. So what they found is that because of this reduction of the metals due to the binding sites, the A-beta can remove hydrogens from neurotransmitters and cause oxidative stress because there's this buildup of ROS, or reactive oxygen species. And so oxidative stress is when there's a buildup of these oxygen species and the cell isn't able to detoxify them. So the brain is very vulnerable to the effects of oxidative stress because of its high oxygen demand. So here's an overview of what we talked about. Protein folding is subject to many errors, and there are many mechanisms in place to help when proteins are not folded quickly enough. Misfolded proteins cause neurodegenerative diseases such as Alzheimer's, which is characterized by beta amyloid plaques and neurofibrillary tangles. A-beta aggregates directly interfere with neurons and neurotransmitters through receptor-mediated toxicity and oxidative stress, and it causes toxicity in the brain. Any questions? Comments?
Pretty strong. Why do those different, do you know why those diseases have an overlap in the proteins that cause them?
So, no. However, I mean, Alzheimer's and Parkinson's, for example, have, so, okay, wait, let me go back. So these three affect the brain. This is just a separate, different thing. So that kind of makes sense because it affects the cognitive function. So it has similar symptoms between these diseases. So I think just the connection between where these proteins misfold in the brain is the direct correlation between them.
Thank you.
Yeah. So you talked about that one drug, I forget. Memantine.
Memantine?
Yes. So it inhibits the methylphosphate receptors. Are there any other drugs that have been used in Alzheimer's?
So I read a little bit about cholinesterase inhibitors that affect the acetylcholine neurons, and so they're looking at inhibiting the degradation of acetylcholine because that's what's creating the signals. And when, like in Alzheimer's, this acetylcholine is being degraded too early or too much, and so they're trying to find these inhibitors that prevent that degradation of acetylcholine. But yeah, that's what we're also looking at drugs that bind directly to A beta to prevent the interactions between the proteins so they don't aggregate. So I thought that was really interesting, but I'm not sure the names of the drugs specifically.
Can you go to the slide that shows how A beta is formed?
This one?
Yeah,
This
this one. So it seems like this is an intentional pathway.
The amyloidogenic pathway?
Yeah, like our body has evolved this for a reason.
I mean, I guess there wouldn't be a reason why, like, are you saying because of this mutation, this enzyme comes in, like, evolutionary-wise?
Yeah.
I mean, yeah,
And then
Yeah.
also, like, the alpha beta that is acting on neurons is not aggregated, right? So, like, when you're talking about it interacting with neurons, it's not aggregated. So maybe alpha beta has an actual function.
Yeah, so there's the amyloid protein that's, like, normally in your body, but it's the amyloid beta that's the one that aggregates. So these, I'm not really sure if these would be just the peptide or if they're clumped together. So that could be, I understand where you're coming from there. But.
Can I follow that up?
Mhm.
So do you know if beta secretase has another function that doesn't have to do with Alzheimer's?
This seems
I mean, I would
bizarre that
assume
you have
so.
all
Like,
the
it wouldn't.
protein.
I don't know why it would be in our body otherwise. I could Google it. So yeah, that's interesting. I didn't really think about it that way. Like why beta secretase exists, if it's specifically for this mutation on the amyloid precursor protein.
You said that the immune cells kill the neuron. Could you go into the...
Yeah, so, I mean, all I really read was that the immune response was activated, and because these neurons were dysfunctional or not functioning at all because the plaques were blocking the cell-to-cell signaling, because they're not functioning correctly, the immune response is like, this is bad, and it just goes and kills the cell. Yeah.
Actually, can you go to the next slide? Just
Sure.
so we don't like, oh sorry, I guess it's a previous. The one that has the, so the extrasynaptic activity. So does that happen because there's an overexpression of that receptor, meaning like it's more abundant in these conditions than it would be otherwise?
So I'm pretty sure that these receptors normally exist. It's just the fact that A beta activates them and allows the glutamate and the glycine to bind. So, like, they normally have that magnesium that stays there until the depolarization phase. But then, I guess, if you want to consider, like, A beta just, like, like it binds to it and opens up the active sites for glutamate and glycine, which then opens the receptor and allows calcium to flow in.
So it doesn't happen with the ones on the...
No, so it doesn't bind to these, because these will just keep working normally. It's just these extrasynaptic receptors.
So the first one will be different ones, different makeups, different
Yeah.
receptors.
Yeah, I think.
You would know more than I do in this case. 'Cause you
Okay,
prepared for
maybe
this.
I don't understand your question.
So are they different types of channels that we're dealing with here?
Like,
The ones that are right underneath the synapse right there?
no, so they're all the same receptor. They're all the same channels. They all flow calcium through, and they all bind glutamate and glycine in order to open them. It's just the fact that A beta activates these when it's present. If this is not present, then it's just going to be normal.
So it's more of an allsteric reaction with that one then?
Yeah. Mm-hmm.
I have some general questions. So when I asked you to define stress, what I meant was, what would cause stress on a cell? I know that you said like having excess protein is stressful, but I mean, what causes that? Or like, under what conditions would you get this? I know, I can think of things that are like general stressors for cells, but in the case of Alzheimer's, like what is that? Or like, is that not the cause of misfolding? And I don't know. Yeah.
So, okay. The mechanisms that cause protein misfolding, like the change in pH, temperature, things like that, weren't necessarily directly related to Alzheimer's.
Okay.
Alzheimer's is just because of that mutation on the APP, which then causes the A beta to be like technically misfolded because it's not really supposed to be cleaved off of that protein. And then because of its misfolding, it aggregates and forms the plaques and blah, 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 blah.
Well, for the stress thing, I know that for Alzheimer's, lack of sleep actually is one of the big stressors.
Lack of sleep.
Oh yeah, also smoking, right?
Smoking, lack of sleep, and extreme activity.
Sleep's important.
And you could do like crossword puzzles.
Yeah,
Like
stimulate your brain.
Yeah.
If you create new pathways,
Yeah.
then go get around the plaque or the clumps. But if you don't.
Right.
You
That's
have
interesting.
the ER stress.
Sure.
I think the plasma reticulum regulates misfolded protein or breaks it up. So I don't know.
I mean, that's what it says.
Yeah.
Yeah, through this system.
So it would be more misfolded proteins than like it would then cause stress. So like one of the stressors. That just
Right. Like the ER can't keep up with it.
Yeah.
There's just
So
too much
it's
Yeah,
to
like if
handle.
there's a mutation or something, right?
Yeah.
And so toxins technically can cause stress or
Or
sun damage or.
reacting on the species
Yeah.
that
Right.
overwhelms the antioxidant
Right.
production of the body.
So that's more what I was looking for. It's not
Sorry.
stressors. Okay, so can we just talk briefly about the biochemistry of alpha helices versus beta sheets?
Sure.
So like obviously beta sheet formation is one of the common folds in these really aggregated fibrils. So and maybe this is better a question like a discussion for the class, but like what is the reasoning between beta sheets maybe being more stable than alpha helices? And then also, when we look at proteins, and this is a direct question for you, when we look at proteins, do we see more alpha helices than beta sheets in folded proteins? Or, do we feel that they're distributed really evenly? What's the...
From my experience, I've seen that there's more alpha helices, just from the structures that I've looked at for
At least in, like,
cytoplasmic proteins.
Yeah, cytoplasmic proteins is
Yeah. There's always there's a mixture of alpha beta, what we call alpha beta proteins. They just have a little bit of both. I think it's more common to see a purely alpha helical protein than it is to see a purely beta sheet protein.
So isn't that interesting that like when they misfold, it's a lot of beta, and it seems like that's the lower energy form? And I mean, I wonder if it's related in some way to protein turnover and recognition, or if there's like a strategy involved in this kind of thing. So if you go to your first slide that shows the alpha helix, obviously you don't show a beta sheet here because they're just not very common. But do you know how beta sheets fold?
No.
That's not wrong.
Between the
Amino H of one and the carboxyl O of another or something? No, I don't know.
I mean, that's true too.
Alright.
But what do we call those? Those are part of the side chains or the peptide backbone?
Backbone.
Right. So in both secondary structures, the backbone is causing, the hydrogen bonding is within the backbone. When you actually look at an anti-parallel beta sheet, the hydrogen bonds are parallel, right? They're like very straight, whereas here you can see they're like a little bit wonky. I wonder if that is what causes beta sheets to be more stable when folded, or when misfolded.
Because with hydrogen bonds, the more head-on, like parallel they are, the stronger they are. If they're a little askew, they're going to be slightly weaker. So, yeah.
That's how I was interpreting it, because they're straighter.
Yeah.
They're not
And then
wonky.
are parallel beta sheets a little bit askew? And so almost all proteins will kind of naturally fold into an anti-parallel beta sheet because the peptide backbone allows that conformation, which is why that's a really common misfold and like super stable minimum energy.
Right.
So if you have two proteins, like when you were talking about Parkinson's and Alzheimer's or whatever, are they different proteins that are folding these? Or like when you say tau proteins, if you take the tau proteins that are forming in Parkinson's and the tau proteins that are misfolding in Alzheimer's, are they exactly the same proteins? Or
Okay, so I didn't really focus a lot on Parkinson's since I kind of just like dove right into Alzheimer's. However, I do believe that it is the same protein and it's the same kind of misfolding, but they have like different effects. Like the mechanisms that I talked about for A beta won't necessarily be the same for Parkinson's, I believe.
okay.
That's how I interpreted the literature.
I have a question about that, kind of going off. Oh, so Alzheimer's disease is a result of misfolding in amyloid beta and tau, or is it one or the other?
It's both.
Oh,
So
okay.
I focus more on A beta, but the where did I do it? I think one slide here. It's because the tau proteins are normal, like in your body, and they stabilize the microtubules in the neuron. But there's a conformational change because of some misfolding of the tau protein, which makes them no longer able to stabilize the microtubules, and so that's why they start to degrade and fall apart.
Good question, the misfolding. Just to clarify, so this issue with misfolding, is it a post-translational thing? So is the protein folded properly and it's once it gets processed by a protease, that then that product is aggregating?
So I was thinking the same thing when I was like finalizing everything, because I was almost starting to get confused about like, okay, mutation versus misfolding, like the mutation on the gene, like, so I'm pretty sure that it's, I think that it's pre-translational due to like the mutation, and then because of the mutation, it doesn't fold right post-translationally. Does that make sense? Like, I guess if there's a wrong amino acid somewhere, like there's some, like in the chain of amino acids, and it starts to fold, and for some reason like a hydrophobic residue ends up on the outside, like that's why it's misfolded.
I guess what I was thinking, because and this occurred to me while we were going over the slides again, is that that alpha beta protein, it is embedded on the membrane, right, the neurons. So it must have been folded enough to make it into bypass proteasomes or anything, and it was deemed good, if you will, for it to make it to the membrane, and it wasn't until the secretase chopped it up that you start
That's
seeing
what-
issues.
Yeah.
And kind of similar with this one, when you go with tau, and this is not the focus of your talk, it seems
Mhm.
that is a consequence of the hyperphosphorylation.
Right.
So it was
Mhm.
fine until
Mhm.
something that was
Right.
misregulated somewhere else went on it and started phosphorylating it.
Yeah. Yes. I don't know what to answer that question.
Yeah,
Honestly.
It's like a question/comment.
But yeah, yes, it is the hyperphosphorylation of tau which causes the conformational change.
What is the role of the parent protein of A beta?
What do you mean?
Like, A beta is cleaved off the protein,
Mm-hmm.
right? Why does it make that original protein? Yeah, like what is
Why does
APP?
Amyloid precursor protein. So this is just a protein that's normally in your body,
Uh-huh.
and I couldn't tell you its function, but it's just all I know is that it's non-toxic, normally in your body, and this is the normal pathway. And the only reason why A beta is formed is because the beta secretase cuts the APP in a different spot than the alpha secretase
So-
normally.
AICD is
Normal,
what is-
non-toxic.
But what is it?
Oh,
I mean, like, one of these parts has to have a function, right?
right.
Is it the piece that's left in the membrane? Is it the APPS alpha?
That has a specific function.
Like a use. The brain doesn't just make a precursor to
Stuff.
Alzheimer's for fun, right?
Right.
Like, it has to have an actual function.
Yeah.
And so I just wonder, I mean, like, yeah, this seems really evolutionarily
Mm.
like a poor choice.
Right.
Energetically.
Big oops.
It must have a real function. Maybe we don't know what it is. I guess that's possible. Or maybe P3 has a function.
I don't know, they just call it P3.
Yeah, precursor 3.
Yeah.
And then, but I mean, like, also the fact that A beta binds to copper and iron and can serve as a reductase, like, that indicates to me that there is some actual function. That is just, like, really poorly
Yeah.
constructed.
And it activates ion channels, so maybe it's supposed to activate something else under normal conditions. Although you only get that with the mutant forms.
Mhm.
I had a biochem question.
Awesome.
I mean, not that these are not biochem questions, but do all proteins fold in the EO?
No, in the cytosol cell?
Which ones fold in the cytosol?
The ones that go to the membrane? I don't know.
The other way around.
Oh.
So the ones that are meant to be in the membrane are taken to the ER, and then they're
Oh,
embedded
right.
onto the membrane of the
Mhm.
ER, and they're shipped to the
Right.
vesicle, the to the
Mhm.
target membrane. And so are the ones that are to be secreted, like the ones that you have
Right.
synapses, right?
Mhm.
Even though they're aqueous proteins, right? They're hydrophilic. They're enclosed in these vesicles in the ER, so then when they're shipped out of the membrane, they actually are released instead of fusing with the target membrane. All right, this may be my last question, maybe? No, my second to last. So of course, I'm always interested in phosphorylation. So you talked about the pKa phosphorylates A beta. Is it like one phosphorylation?
I
or-
think so, because it said specifically on the serine residue. Yeah, on position 8. So I
Oh,
think it's
just
just
one.
one phosphorylation on that serine
Oh,
residue.
sorry, I missed that.
That's okay, I don't think I
That's-
said it.
That to me seems very surprising, that one phosphorylation event.
Right, I mean, maybe it's indicating like all, well, no, serine residue on position 8.
Expecting something more like tau that was hyperphosphorylated, and I can see that too.
Right.
How big is it? How many positions does it have?
39 to 42.
Okay, so that's a lot.
Yeah.
I think also the plaques determine like the onset of Alzheimer's, so like if they're longer, then you have Alzheimer's sooner. Whereas if you had less than the threshold, it like some people can have just like a low number of plaques and not have Alzheimer's.
Right.
But then once you start accumulating the plaques, you go above that 39 that you get onset. So some mutations might increase the rate at which you get plaques. So it would change the onset when you get younger.
That's a deviation as well.
She also said that like there's like that one main mutation that she
Yeah,
discussed.
the
There's-
valine to isoleucine.
But there's others as well that might
Many
have,
others.
that might have like a characterized for like a earlier onset or ones that don't produce as much amyloid plaques.
Maybe something with a lot of phosphorylations wouldn't aggregate well either. I want to tell one another. Okay, the last question that I'll ask, because you brought up dominant negative.
Mm-hmm.
So are both proteins there, wild type and mutant, dominant negative?
Yes. Okay, what I understood was that so you could have like different types where you have one of each or then you have both. And so like, I think it was like the 25% and the 50% come from like those Punnett squares from genetics, which I'm taking that next semester, so I'll learn. But yeah, I believe you can have either option.
So the one with the mutation takes over.
Right.
That's what
It
is
makes it-
dominant.
Right. Overall non-functional.
I want to know, is amyloid plaque accumulation implicated as, for sure, definitely the main cause of Alzheimer's?
Yes.
It's 100% implicated?
Well, yeah, it's that plus the hyperphosphorylation of tau.
Okay.
I've seen a thing that one treatment plan for Alzheimer's is sonicating your brain
That sounds,
to like break apart the
plaques.
plaques. Yeah, so this is like a strategy, like ultra focused sonication, because you can get like overlapping waves, so you could like really focus on the specific plaques, and that would break them up, and then
Oh
you'd
my
not
God.
get rid of them.
That makes you
I don't know. I have no idea. I don't know what the
You're-
progress
you're
is on that. I
good part
just
of your
heard it.
brain.
Like, I use a sonicator, like
Yeah,
a sonicator in my lab, and so like it
That's
would be
insane.
amazing.
To break open bacterial cell walls. Just like
I mean, like
Oh my
the one
God.
that we have will cut DNA, genomic DNA, down to 500 base pairs, sections.
That's what we use it to.
It's like amazing.
Oh my God.
Yeah.
That's crazy.
We had in genomics class, we had to read some papers about this, and like one of the treatments that we're proposing was to like make a way to like cut the plaques at certain intervals so that they can't form the 39, like the threshold of repeats. So that like if you did that, then the person wouldn't have Alzheimer's. If you like,
Found like a specific restriction and
or like develop something that
Proteins.
just like only cut in certain like parts.
cool.
CRISPRize.
Yeah, CRISPR.
Great, let's call it. Hopefully I made it. Who's on Monday?